Hey everyone, it's Ms. Lopez. I'm going to talk about how to predict substances moving across the membrane using the 5 processes we've talked about in class. Here we go. The first is simple diffusion. Remember that this does not require energy. Substances are going to move down their concentration gradient, and this is going to be possible only by substances that are non-polar, because they have to cross the non-polar or hydrophobic area of the membrane. The substances will also need to be small. For facilitated diffusion, substances are still not going to require energy, and the movement is going to be down the concentration gradient. The difference is now we need a protein of some type to allow the substances, which are going to be small and polar, to cross the membrane. This is the part that gave students quite a bit of trouble, determining whether or not something was polar. If you think back to when we talked about the properties of water, or when we talked about how cholesterol plays a role in the membrane, there are 2 things you can look for to help determine if something is polar. The first is, does it have a charge? Remember that when we talked about the properties of water and the structure of water, we talked about how the oxygen takes the electrons from hydrogen away, creating a slight positive and slight negative charge. Because of that slight negative and positive charge, we described water as a polar object. So you can look at a substance and see, does it have a positive or negative charge to determine whether or not it's polar. The second thing you can look for is that hydroxyl ion, or that hydroxyl group we had back from properties of water. So even if a substance doesn't necessarily have a charge written on it, you can look to see what that ratio of oxygen and hydrogen, or that hydroxyl group, is to determine if that section or if that molecule is going to be polar. The next type of transport is active transport. With active transport, the key thing that you're going to look for is that energy is required and that substances will move up their concentration gradient. Active transport will always require a protein to assist in this process, because it's the protein that's going to need that energy in the form of ATP. Next, endocytosis, which brings large objects into the cell. If an object is a bacterium or something of that size, it is called phagocytosis. If it's a liquid, it is called pinocytosis. Last is exocytosis, which is also involved with large objects or large amounts of objects, but these substances will always be removed from the cell or released from the cell. The process of osmosis is an example of facilitated diffusion, so we are not going to include it in one of these or one of the 5 processes, because it is a subcategory of facilitated, much like phagocytosis is a subcategory of endocytosis. Thanks y'all so much for watching. I hope this helped.